Hello everyone, welcome to PG Pathshala. In this video, I'll be covering Module 7 of Cellular and Molecular Biophysics. This module is on aging and longevity. I am Ashik Siti and currently I work at the All India Institute of Medical Sciences, New Delhi. At the end of this module, you should be able to explain Hayflick's limit, different biochemical mechanisms of aging, the mechanism of telomerase, various causes of premature aging, and biomarkers of aging. Let's start by answering the question, why do we age? In simplest words, the answer is we age because our cells age. It has been observed that cells from different tissues fail to divide more than 40 to 60 times. This limited potential for somatic cells to undergo mitosis, that is cell division, is known as Hayflick's limit. Who was Hayflick? Leonard Hayflick was an American anatomist. Before Hayflick, it was believed that normal vertebrate cells have a limitless replicative potential. But in 1960s, Hayflick conducted a series of experiments to disprove this hypothesis. Let's see what Hayflick did. Hayflick maintained in vitro cultures of normal male human fibroblasts that had undergone a large number of cell divisions, like 49 times. He did this along with maintenance of normal human female fibroblasts that were relatively young, like those that have undergone up to 13 cell divisions. When the cultures were examined, after a time duration required for some 17 cell divisions or so, he observed that only female fibroblasts were present and not the male ones. Thus he concluded that fibroblast cells fail to survive after a certain number of cell divisions. We will now see some mechanisms that contribute to cellular senescence. For example, we will discuss about shortening of telomeres, DNA and protein damage, nutrient sensing, and epigenetic mechanisms that lead to aging. Telomeres are repetitive sequences that are present at the end of linear chromosomes. If you see this diagram of two chromosomes that are joined at the centromere, you can see those red regions that are located at the end of the chromosome. These are called telomeres. A chromosome is nothing but a condensed molecule of DNA. Because of the linear nature of the eukaryotic DNA, there occurs a peculiar phenomenon in these chromosomes during DNA replication. This is known as Replication Problem. With progressive cell divisions, the telomeric ends undergo attrition and this leads to cell cycle arrest. This image shows DNA replication. During replication, RNA primers are synthesized by the primase activity of DNA polymerase alpha. The leading and lagging strands are synthesized by DNA polymerase delta. Note that DNA polymerase delta can add nucleotides only to a pre-existing nucleotide oligomer. Leading strand is synthesized in a continuous manner while the lagging strand is synthesized discontinuously in the form of Okazaki fragments. Thus, DNA replication is semi-discontinuous. Once DNA is synthesized, the RNA primers are degraded by enzymes like RNase H1 and FEN1. DNA polymerase delta now fills these gaps due to its polymerase activity and the nicks are sealed by DNA ligase. Since there is no pre-existing nucleotide oligomer at the 5' end of the newly synthesized leading strand, DNA polymerases cannot fill the gap here. This leaves a 3' overhang at the end of the chromosome with loss of DNA at the 5' end. The same happens at the 5' end of the newly synthesized lagging strand, also due to the same mechanism. With progressive cell divisions, the telomeric ends suffer attrition and this leads to cell cycle arrest. Certain eukaryotic cells express an enzyme called telomerase to counter this replication problem. Telomerase is a reverse transcriptase. That is, it has RNA-dependent DNA polymerase activity. And the intriguing feature of telomerase is that the RNA template which is required for DNA synthesis is present within the enzyme itself. Thus, telomerase is a ribonucleoprotein. The expression of telomerase is highest in germ cells, followed by stem cells. Telomerase is also expressed in cancer cells. Let's see the mechanism of action of telomerase. Human telomeres are rich in the repeating AGGT sequence in the 3' end. The RNA template present in the telomerase is complementary to this sequence and extends beyond this unit. Thus it can bind to the 3' end of the chromosome and synthesize new DNA at the 3' end using its own RNA as template. Telomerase then translocates to the 3' end further and continues this process, extending the 3' end of the chromosome. The purpose of this extension of the 3' end is that a new RNA primer can now bind to this strand. DNA polymerase now adds nucleotides to the newly bound RNA primer, thus the 5' end of the newly synthesized strand is restored. As the ends of the chromosome have now been sufficiently extended, removal of the RNA primer by ribonucleases won't affect the chromosome integrity even though it leads to a 3' overhang. And this 3' overhang usually invades the intact DNA duplex to form a T-loop, which is further stabilized by proteins like TRF1 and TRF2. And this T-loop protects the chromosome ends from nucleases. In somatic cells, telomerase is either absent or is expressed at very low levels. Studies in mice have shown that absence of telomerase in the animal as a whole leads to accelerated aging, which is manifested by stem cell depletion, tissue atrophy, and organ failure. Reactivation of telomerase enzyme in experimental animals resulted in elimination of degenerative phenotypes and restoration of near normal phenotypes. If this is the case, why can't we use telomerase to prevent aging of somatic cells and thus the aging of the organism as a whole? At this point of time, the answer is no, we can't use it. This is because, apart from germ cells and stem cells as I previously mentioned, cancer cells consistently express telomerase. Telomerase expression is responsible for replicative immortality, which is one of the hallmarks of cancer. But we can use this knowledge. Telomerase is used as a biomarker for diagnosis and prognosis of certain cancers, and certain inhibitors of telomerase, for example imetelstat, is already in clinical trials for myeloproliferative neoplasms and myelodysplastic syndromes, which are pre-malignant conditions. Moving on to the next mechanism of aging, that is DNA and protein damage. DNA and protein damage can be induced mainly by two mechanisms. One is free radical mediated damage and the other one is radiation mediated damage. Free radicals refer to those chemical species with an unpaired electron in its outer orbital. Closely related but different species include reactive oxygen species and reactive nitrogen species. The former includes superoxide, hydroxyl, and hydrogen peroxide, while reactive nitrogen species include peroxynitride radical and nitric oxide. As you can see in this figure, not all reactive oxygen species, or for that matter reactive nitrogen species, contain an unpaired electron in their outer orbital. Thus, all ROS and RNS are not free radicals. However, even those ROS and RNS that are not free radicals can generate free radicals and are deleterious. These molecules are highly active due to unpaired electron in their outer orbital and can react with a wide variety of molecules in the cells like nucleic acids, lipids, proteins and other compounds. Biological oxidation is a major source of free radical generation. NADH and FADH2 donate their electrons to the complexes 1 and 2 respectively of the electron transport chain, which is located in the endomitochondrial membrane. These electrons are transported along complex 3 to complex 4, where oxygen, the final electron acceptor, accepts the electrons and gets converted to water. The efficiency of this process is diminished in older mitochondria, which leads to electron leaks at various complexes. Normally, a single molecule of oxygen accepts 4 electrons and 4 protons in the complex 4 to generate 2 molecules of water. However, if there is mitochondrial electron leakage, oxygen may end up accepting 1, 2 or 3 electrons, leading on to formation of superoxide, peroxide or hydroxyl ions respectively, and hydroxyl ion is one of the most potent free radical known. Apart from electron transport chain, the other sources of free radical include prostaglandin synthesis, phagocytosis, in which enzymes like NADPH oxidase and myeloperoxidase leads to free radical generation, inflammation, xenobiotic metabolism, and free radical generation mediated by transition metals like iron and copper in reactions like Fenton reaction. How does free radicals lead to cell injury and aging? It can be because of DNA double-strand breaks, single-strand breaks, formation of DNA adducts, etc. DNA adducts can be hazardous because certain oxidized bases and nucleotide adducts are identified different from their parent bases by the DNA polymerases. This leads to introduction of mutations in the daughter strand during replication. In presence of free radicals, the DNA repair mechanisms that are present in the cell are overwhelmed, and alterations in DNA can lead to cell cycle arrest and senescence. Free radical induced damage can affect protein function and can enhance protein degradation. Protein adducts also leads to abnormal protein folding. When unfolded proteins accumulate, the cell resorts to a salvage mechanism known as unfolded protein response. In unfolded protein response, the cell tries to protect itself. How does it do so? It can cause a generalized reduction in translation, leading on to decrease in synthesis of proteins. It can overexpress chaperones that will help in folding of the unfolded proteins. It can translocate unfolded proteins to the endoplasmic reticulum for refolding. It can also induce proteasomal degradation of unfolded proteins. If nothing works, the cell will resort to phenomena like apoptosis, which will result in cell death and thus loss of cells, ultimately leading on to aging. Free radicals can also react with lipid components of cell membrane and other biological membranes like polyunsaturated fatty acids, and it can lead to widespread membrane damage. They also can oxidize lipid components of molecules like low density lipoprotein LDL to form oxidized LDL, which is highly atherogenic and which can lead to atherosclerosis, further leading on to cardiovascular diseases. Radiation mediated damage. UV radiations of the wavelength 200 to 280 nanometer, that is UVC, is the most harmful of the UV rays, but these are usually filtered by the ozone layer. UVB is biologically active but cannot penetrate beneath the superficial skin layers. Exposure to UVB can predispose to skin cancers. UVA can penetrate beyond superficial skin layers and is responsible for aging of skin. Exposure to harmful UV rays can cause direct DNA damage in the form of pyrimidine dimers and chromosomal instability. What happens to the pyrimidine dimers? In prokaryotes, pyrimidine dimers are corrected by an enzyme DNA phosphorylase, but this enzyme is absent in eukaryotes. Eukaryotes rely mainly on nucleotide excision repair for removal of pyrimidine dimers. In a condition called xeroderma pigmentosa, the patients have defective nucleotide excision repair and they are prone to skin cancer and premature aging. The other pathway by which eukaryotes handle pyrimidine dimers is a specific form of DNA replication known as translesion DNA synthesis, which is catalyzed by specific DNA polymerases like DNA polymerase eta, which is an error-prone DNA polymerase. Defect in DNA polymerase eta leads to a variant of xeroderma pigmentosa. Moving on to the next mechanism of aging, this is related to nutrient sensing. Sensing of a particular nutrient is mediated directly by the molecule binding to its putative sensor or indirectly due to surrogate molecules downstream the nutrient metabolism. Examples of sensors include GLUT2 and glucokinase, which are sensors for glucose, SCAP protein, which is SREBP1 cleavage activating protein, which is a sensor for glucose, adipokines like lectin, which is a sensor of lipid storage, and AMP activated protein kinase, which senses energy balance. An increase in carbohydrate and protein intake is associated with an enhanced signaling involving insulin IGF-1 signaling pathway. Signaling through this pathway leads to an increased generation of mTOR, which is nothing but mechanistic target of rapamycin. Some people call it mammalian target of rapamycin. And mTOR is associated with inhibition of autophagy, which is required for clearance of damaged and misfolded proteins. Studies have shown that an inhibitor of mTOR, that is rapamycin, can relieve symptomatic effects of aging, probably by promoting autophagy. Similar effects are postulated to be exhibited by inhibitors of insulin signaling like metformin, a very widely used oral hypoglycemic agent. Another class of proteins associated with nutrient sensing is sirtuins. Sirtuins are NAD-dependent protein deacetylators, and these proteins form a link between nutrition status and gene expression. Deacetylation by sirtuins has been shown to activate proteins like isocitrate dehydrogenase, And isocitrate dehydrogenase activation can lead to an increased production of NADPH, and this further activates NADPH dependent antioxidant systems which can scavenge free radicals, thus prevent aging. Sirtuins have also been shown to regulate histone deacetylation, and thus it can lead to alteration in transcription. Resveratrol, which is present in red wine, is a sirtuin activator and is a potential candidate for anti-aging therapy. Let's now see the role of epigenetics in aging. Epigenetics refers to the heritable changes in the genome without any change in the sequence of DNA. Epigenetic mechanisms play a prime role in regulation of gene expression. The histone deacetylation action of sirtuins, which we just discussed, is one example of an epigenetic modification. The other examples being other histone modifications and DNA methylation. DNA methylation is a major epigenetic mechanism which has implications on aging. 5-methylcytosine, an epigenetic modification, mostly exists in DNA in the context of CpG dinucleotides, that is a C and G placed together, and if you see, almost 70 to 80% of all the CpGs are methylated, and CpGs are commonly found in the 5' end of the genes. And the methyl group of the 5-methylcytosine can protrude to the major groove of the DNA, and it can interfere with the binding of various transcription factors. Aging has been found to be associated with an alteration in the CpG methylation pattern, and this process is known as age-related methylation drift, with an alteration in the methylation patterns across promoters and heterochromatin. This can lead to alteration in gene expression, and this may be associated with aging. Let's now consider some pathological states that are associated with premature aging. We have already seen that defective DNA repair, as in nucleotide excision repair, and translesion DNA synthesis by DNA polymerase eta leads to premature aging in xeroderma pigmentosa. Not surprisingly, a battery of conditions can lead to premature aging, most of them involving defective DNA repair. This table lists some conditions associated with premature aging like Fanconi's anemia and ataxia telangiectasia and Bloom syndrome, and if you see, all these conditions involve defective double-strand DNA repair. Another well-studied and well-characterized premature aging syndrome, Hutchinson Gilford progeria syndrome. The protein defective in this condition is lamin, and lamin is a component of intermediate filaments that forms a nuclear lamina. The defective protein lamin is also known as progerin, is a truncated form of lamin, a precursor protein, that is prelamin, and this is usually farnesylated. Normally, the farnesylated portion of prelamin A is cleaved by proteases to form lamin A, but a mutation in that site leads to loss of site. The protein defective is lamin, which is a component of intermediate filaments that form the nuclear lamina. The defective lamin protein is also known as progerin, is a truncated protein of lamin, a precursor protein, that is farnesylated. Normally, farnesylated portion of prelamin A is cleaved by proteases to form lamin A, but mutations lead to loss of the site at which the proteases can act. Defarnesylated prelamin A accumulates in the nuclear membrane and disrupts normal cellular function. Inhibitor of farnesyl transferase, lonafarnib, has been found to be useful in ameliorating the manifestations of Hutchinson Gilford progeria syndrome. Let's now see biomarkers of aging. Biological age of a person might be different from his chronological age. Since biological age is a risk factor for various disease conditions, including cardiovascular diseases and cancer, biomarkers that reflect biological age can have various clinical utilities. Further, these biomarkers can also be used in assessing the effectiveness of any intervention that can prevent aging. Biomarkers of aging include telomere length in leukocytes, which can be measured by terminal restriction fragment analysis. We'll see that in the subsequent slide. Telomerase activity determination by TRAP assay, phosphorylated histone protein H2AX assay, which is a marker of DNA repair, age-related changes in DNA methylation, which we previously discussed, assessment of non-coding RNAs that are related to aging, assessment of molecules that are related to nutrient sensing, and measurement of advanced glycation end products. So we will see few of them in detail. In terminal restriction fragment analysis, it is nothing but a modified southern blotting and utilizes restriction enzymes that does not cleave the telomere sequences. So you can use telomere specific probes after restriction, digestion, electrophoresis and blotting of DNA fragments to determine the telomere length. Advanced glycation end products, also known as AGE, are formed due to non-enzymatic addition of carbohydrates to proteins, lipids and nucleic acids. Accumulation of AGE is associated with age-related disorders and cellular dysfunction. We can measure advanced glycation end products by specific assays or by using chromatographic techniques like high-performance liquid chromatography or gas chromatography. Evolution is associated with selection of characters that favor survival. But if you see, aging is associated with progressive deterioration of various bodily functions. So it is sort of contrasting. Why should we age? Because evolution should not favor aging. Isn't it so? Not exactly. A limited lifespan actually limits draining of resources. And the purpose of evolution, if you see, is the maintenance of genes that are beneficial for survival. And this is achieved usually during the reproductive years. So all the traits that prolong life beyond the reproductive age will not have any positive selection pressure on evolution. We are at the end of this module. To summarize, somatic cells have limited replication potential, which is known as Hayflick's limit, and the main reason responsible for this is the shortening of telomeres. Certain cells like stem cells, germ cells and cancer cells exhibit the expression of an enzyme called telomerase, which maintains a telomere length. DNA and protein can be damaged by free radicals and UV radiations. Excess calories and nutrients can alter nutrient sensing mechanisms to accelerate aging. Epigenetic alteration and age-related methylation drift is associated with aging. There are many premature aging syndromes and most of them are characterized by defect in DNA repair mechanisms. Different biomarkers are available for identifying the biological age and they might find use in assessing the true biological age and also potency of anti-aging therapies. Thank you for your patient listening. And one final thing, if you really want to live after you have died, donate your organs. Thank you.